Picking up where we left off, steroids are a special kind of lipid that you will find in your body. They are naturally produced. That is always going to have this shape of ring structure in it somewhere. So all the different steroids that you have in your body, all the different kind of hormones and things like that that are based on steroids, they're going to have this shape somewhere. So cholesterol, for example, you can see the shape right here. There's other stuff attached to it, but that basic shape is there. Same thing here, testosterone, estrogen, all these things are going to have this basic ring structure in it somewhere. Things like this, cholesterol especially, kind of gets a bad name for itself sometimes. But we do actually need cholesterol in our body. The bad thing is we don't need too much cholesterol. Like, our body will produce the cholesterol we need. We shouldn't be, eating food that's 90% cholesterol or anything like that. So our body can get the cholesterol it needs and use it to produce the things like testosterone and estrogen that it needs. Moving on, let's talk about proteins. So proteins are essentially the molecular machines that make life in the cells happen. So structural components like muscles, skin, and cartilage are made of protein. Proteins transport oxygen in the blood. They act as antibodies. They function as hormones to regulate your metabolism. Proteins are one of the biggest keys to life. Proteins are made of lots and lots of amino acids. They're actually polymers of amino acids. They're very, very different than a typical polymer. So if you just walk up to a random chemist and ask them to describe a polymer, they might not describe a protein because the different sections of the protein are actually quite different. But they are technically considered a polymer. These amino acids that the proteins are made of are always going to be composed of a carbon with a hydrogen attached to an NH2 group, the amine group, and a carboxylic acid group. So this section of the molecule doesn't really ever change. The thing that's key is this bit. This is the group attached to this specific amino acid that gives that amino acid its character. The rest of the molecule here is mostly just used for linkage. So this amino group will match up with a carboxylic acid group on a different amino acid to produce what we call a peptide bond. And so this peptide bond is what holds the protein together. And then the specific order of these different groups on the different amino acids is what gives the protein its character. So it decides what it's going to do, if it's going to do anything at all. It could just be used as something that just sits there and is a structural protein. Or it could be a protein that actually does some kind of work, that has a job. It just depends. So there are lots of amino acids. So I have a list of them here. And again, I'm not a biochemist. I don't think this is a complete list, but I could be wrong about that. Again, not a biochemist. But for sure, these are the most common amino acids, at the very least. You have to memorize all of these, including how to draw them. Just kidding. I know, I gave you a heart attack there for a second. You do not have to memorize them or how to draw them. But I would encourage you to give them a once-over, especially if you're going into some kind of medical field, because you will probably be tested over them in different class. They will probably come up in your biology classes if they haven't already. So we have lots of different amino acids here with varying sizes and chemical potencies of the R groups. So some of these have really simple, really small R groups, like glycine. It's just a hydrogen. Super boring. Some of them have much bigger R groups, like arginine here, which is the pirate amino acid. That's the way it was explained to me when I was taught. Pirates like arginine. Ha 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 ha. But a much bigger group, a lot more chemistry happening here. 3 different nitrogens in this amino acids R group. So very, very different chemistries from one amino acid to another. So when we take a string of amino acids and bond them together to form the most basic part of the protein, the string of the amino acids will twist and fold into very specific shapes. And these shapes are just as specific as the actual sequence of the amino acids themselves. And there are 4 different levels of shapes for proteins. So it's not just the order of the amino acids, it's how they fold together that are important. So we have primary structures, secondary structures, tertiary structures, and then quaternary structures. So primary structures are just the order of the amino acids. It is literally just the simplest structure itself. It's just how are the amino acids lined up. So here we have alanine, glycine, I forget what, aniline maybe? I forget. Again, not a biochemist. But we have a string of amino acids here. That is the primary structure. It's just what is the order of the amino acids. The secondary structure is then how do you take that one string and fold it. Its immediate structure past just the order of the amino acids. So that could be in the form of pleated sheets, or it could be in the form of a helix of some kind, or it could just be random, just depending on what protein you're making. And we're going to take a slightly closer look at some of these in just a second. The tertiary structure is then saying, okay, we took this, we formed it into an alpha helix. Now, let's take the alpha helix and make it into its own shape. So it's going to have a 3rd level of structure, a 3rd level of shape. And then a quaternary structure is taking a completed peptide chain and matching it up with other chains. So we have, I believe in this picture, 4. It looks like there's 4. There's one there, one here, and one back there. 4 different peptide chains that have all come together to form some larger structure to do something. And so a quaternary structure actually goes beyond a single chain. It's multiple chains coming together. So again, primary structure, just the order of the amino acids. Secondary structure, how that string of amino acids is folded. Tertiary structure, taking the entire string and forming it into some shape. And then the quaternary structure is taking multiple strings to form into a shape. So I said we'd come back to look at a few of these different shapes more specifically. So first of all, here we have quaternary structures. This is an example of a hemoglobin. So hemoglobin is actually made of 4 different polypeptide chains. Each of those individual polypeptide chains has a heme group in it that has an iron in it somewhere. There's an iron atom in that heme group that helps it to carry oxygen throughout the body. And so we have 4 different locations to actually carry oxygen in the hemoglobin. Each hemoglobin group can carry 4 O2s throughout the body, essentially. So that is an example of quaternary structure where we have multiple strings working together to serve one purpose. Secondary structures, again going back to the previous slide there, this is the immediate structure past the order of the amino acids. So we can have alpha helices, which is not the same as a double helix like you see for DNA, because a double helix would be 2 separate strings, and this is just one string. It is just a single string of molecules, but it is wrapped around in the same shape as a double helix. And so what happens here is, as the amino acids wrap around, what holds it together are actually strong hydrogen bonding interactions is what they're called, although we haven't talked about those in this class. But there are hydrogen bonding interactions between molecules, between specific atoms in different molecules here. So, well, actually, technically it's all one molecule, but you have a CO group here, and that has a very strong interaction with the hydrogen that's attached to a nitrogen over here. And so there's a very strong interaction there that actually holds the structure together, because otherwise it would never do this. Like, there has to be some kind of interaction to hold the structure together. Molecules don't really like being bunched up like that, but if there's a strong attractive force there, it can stay together. Pleated sheets, or beta pleated sheets, are another way of arranging a structure. It doesn't look nearly as pretty as an alpha helix, but it is still very common to find pleated sheets like this. And so with these types of things, you have the same chain, but it's kind of hard to tell, but it does kind of go along, and then it comes back around, then it comes back around. And so it's not a nice, circular structure like the alpha helix, but it is still the same sheet interacting with different parts of itself. So it arranges itself so that, again, oxygens over here can interact with hydrogens over here. And this is also very, very stable, these beta pleated sheets. Tertiary structures, so that's the third tier, if you will, of structures. Not entirely sure why I have them out of order in my slides here, but anyway. Tertiary structures are usually governed by really strong interactions between different parts of the structure. Sometimes it's not quite as strong, but it often is held together by strong interactions. So, for example, disulfide links are by far the strongest interaction that you can have, because here you actually have a covalent bond holding two parts of the structure together. You have a sulfur-sulfur bond that is very difficult to break. The formation of disulfur links, or disulfide links, is actually what makes eggs get rubbery when you cook them. There's a disproportionate amount of sulfur in those egg proteins, and so as you cook them and they start to form these disulfide links, the heat isn't enough to break them, or is not enough to break them, and so it stays solid from all the disulfide links that are formed. But you can have other interactions as well that cause the protein to take that tertiary structure. So you can have what we call salt bridges, where there's an ionic interaction. So you can have negative and positive ions that interact very strongly. You can have strong hydrogen bonds that hold things together. This hydrogen bond is going to be slightly stronger than a hydrogen bond between a nitrogen and a hydrogen interacting with a CO group. So this is just a slightly stronger version of that same type of interaction. And you can also have hydrophobic interactions as well that keep things together, although those are not quite as strong. Those are easier to break. Alright. Last thing here, nucleic acids. So we just learned about how the body has these proteins that can do all kinds of amazing things, but how does the body know how to make them? How does the body know how to make these proteins? Well, the reason that the body knows how to make them is it has excellent instructions, and that is deoxyribonucleic acids, or DNA, or ribonucleic acids, depending on what you're trying to make. So DNA is the instructions that the body uses to make all these different kinds of proteins. Every cell of your body has the instructions to make your entire body, if you believe it. That's kind of crazy to think about. But the DNA, deoxyribonucleic acids, are, again, a polymer, kind of similar to the proteins. But they're made of slightly different stuff. So they're not made of amino acids. Instead, they're made of these deoxyribose molecules that are connected by phosphate groups. And then on the deoxyribo, there is a base, some kind of base attached to it. And there are 4 of these bases. There is adenine, cytosine, guanine, and thymine. Those are the 4 possible bases attached to the chain here, the DNA chain. These 4 bases can only pair up one of 2 ways, though. So if you're familiar with how DNA has that double helix, where you have the two chains of the DNA that match up together, you're never going to have a cytosine base paired up with an adenine base. That's never going to happen. The C and the G are complementary, and the adenine and the thymine, the A and the T, are complementary. So they match up together. They fit each other, essentially. So when DNA is folded into its double helix, the A and the T are always paired, and the C and the G are always paired. And so it's going to look something like this. So you have your double helix here, 2 different strands of DNA, and how they're paired up is using the A and T go together, C and G go together. And they actually fit together because of intermolecular forces between the two. The molecules fit together. There's a strong interaction between A and T. There's a strong interaction between G and C, and they don't really fit to do it a different way. G and A just don't fit together. And this is actually really important for copying our DNA. Do I talk about that here? Yes. Okay. So when we're copying our DNA, it's actually really, really crucial that we have both strands here, and that they be complementary to one another, that one side matches the other side, just the opposite. So again, A and T, G and C, because when we want to copy the DNA, which we do all the time, right now your body is copying your DNA, I don't know how many trillions of copies it makes per minute, but you're constantly making new cells, you're constantly fixing things, making new proteins. Your body is constantly reading your DNA and making copies. And so to do this, what it does is it will split the DNA apart, which, well, get to that in just a second. So it splits the DNA apart, and it reads one side of the DNA, and because it reads the one side, it knows what the other side has to be, because again, they're complementary like that. And so just whatever fits to the other side, it makes that, and so what you get out the other side is now you have 2 complete copies of the original DNA, because they're always paired up like that. So that is in a very, very big nutshell, explained by a non-biochemist, how your body copies DNA. So then to use that DNA, by the way, to produce something, let's say that the reason that you split this DNA apart was not necessarily because you're splitting the cell to make a new cell. Maybe you needed the information in that DNA. Maybe that DNA had the information to make hemoglobin. Your body needs to make some new hemoglobin, and so it's going to take the DNA there, and it's going to copy that DNA. So what it's going to do, you take the DNA and you make mRNA from a section of the DNA in the nucleus. The mRNA, which again, if you really need to know what this stuff is, you'll, I'm sure, talk about it a whole lot in some of your other classes. But the mRNA is read by a protein called ribosome, and a new protein is created using amino acids. So you have this ribosome that comes along here, and it literally reads the DNA as it goes along, and as it's reading the DNA will tell it, you know, this amino acid, then this amino acid, then this amino acid, and so it will literally match up what the DNA is saying to the protein that it is building, and then it will spit out the completed protein out the other side. It's actually really, really cool the way that all those things work. Proteins really are just molecular machines. It's pretty amazing to think about. All right, I don't think this link actually works anymore, but if you want to take a look at an artist's rendering of what happens inside of the cell. What's the name of that video? Well, I can't think of it off the top of my head. If I can think of it later, I might send out an email about it. But there's a video that was made back when I was in college, so a while ago, where they had this artist rendering of all these different amazing things happening in cells. It might be like life inside the cell or something like that. But anyway, I would encourage you to watch that if you are interested in it. But other than that, that is it for this chapter. I know that I kind of rushed through that, partially because, we're at the end of the semester, and partially just because, like I've said, I think a dozen times now this video, I am not a biochemist. This is not my strong suit. And if you really need to know the biochemistry, I'm more than happy to just give you a brief introduction here. But if you really need to know the in-depth stuff, you will have multiple classes over that throughout your college career. So please let me know if you have any questions about that. Good luck with the homework, and I will talk to you all later.